Okay, so hello everyone. Today I'm going to introduce my research. It's called mbImpute, an accurate and robust imputation method for microbiome data. So first, I want to talk about some of the learning materials. So here in UCLA, I would recommend the Math 170A and the Math 170B for you to learn some basics about probability. And for the linear algebra part, I would highly recommend the MIT Open course by Professor Gilbert Strange. And for linear regression with penalization, you can search for the Stats 203 in Stanford.
And let me just make a small correction. The professor's last name is Strange. There's no E in the end, so it's not Strange.
Oh, sorry. I apologize for that. Yeah. Okay, sorry. Yeah, but you can search it on the MIT Open course website. And also for the optimization, you can check the Math 164 in UCLA. And for the coding, I have been using in my project mainly R, and there are some Python code that need to compile for other packages. And you can learn R in Stats 20 in UCLA. And for the biology part, I basically learned from the related papers. And I would think the Google search, YouTube, Google Scholar, and the online courses are all very good materials for you to learn about the related topics in your future research. So first, I will introduce some of the background. So the microbiome studies explore the collective genomes of microorganisms that live in a certain environment. The main groups of these microorganisms include prokaryotes, or called bacteria, the archaea, fungi, and viruses. The number of microbial cells on human body is estimated to be 10 times the total number of human cells. So you can think of the microbiome study as basically the study of bacteria in some certain environment. And for the human microbiome study, they usually study the bacteria living in the human body, for example, in the human gut. And so the importance of microbiome study is that the collective genomes of microbes on human body constitute an extended human genome that encode genetic and metabolic capabilities that humans do not inherently possess. And microbiome plays important roles in diseases such as obesity, diabetes, and some kind of cancers. And the human microbiome can be new therapeutic targets or biomarkers for disease prevention and diagnosis. So that's kind of the incentive for scientists to study the microbiome. So why do we need an imputation method for microbiome data? There are actually a lot of zeros in the microbiome sequencing data. There are 3 types of zeros in the 16S rRNA sequencing and the whole genome sequencing. And I think previous talks have already introduced the single cell sequencing, and maybe you already had some knowledge about what sequencing is. And so here I want to introduce that there are 3 types of zeros. So first is the biological zeros. So those are the true zero abundances of non-existence of taxa in samples. And there are also technical zeros due to the artifacts in sequencing experiments, and the sampling zeros due to limitation in the sequencing depth. And the whole genome sequencing, although do not need to, it's a kind of different from 16S RNA, because this you can see is the RNA sequencing, and the whole genome sequencing is basically DNA sequencing. However, it still has a lot of zeros because it suffers from the host DNA contamination. For example, in the human microbiome studies, the human DNA will have a large proportion in the sequencing. So if we just care about the microbiomes, we need to get rid of those DNAs. So however, because there's a fixed number in the sequencing depth, so this will lead to like more zeros in the microbiome abundance. And the goal of our method called mbImpute is to recover the likely non-biological zeros in microbiome data and reduce data sparsity. Here, the sparsity just means there are a lot of zeros in the data. And then to output an imputed data matrix we want to output an imputed data matrix in the same format as the input data matrix. And the third thing is we want to facilitate the downstream analysis that cannot accommodate high data sparsity. Again, this is like all the problems are caused by the large number of zeros existing in the data. So here to make you aware of what the data look like. So basically it comes with 3 matrices. So the first one is the taxa abundance matrix. So here I will use taxa or taxon to indicate you can think of it as just some bacterial species, and the count reflects the relative abundance of that bacterial species in human body. And so in each row, each number indicates one. You can think of it as one patient, for example, in the human microbiome data. And you can see for those same people or same patients, they have their covariates. You can read from this table that we have the study condition. So whether they have a certain cancer or they are healthy people, and their age, their gender, their body mass index, and other information related. And other than that, we also have the phylogenetic information. So the phylogenetic basically captures the evolutionary relationship among different bacterial species. And so let me go to, so here you can see they are all integer numbers because it is derived from a structure such like this. So this shows like how bacterial species have been separated based on their similarities in evolutionary similarities. And so for those bacteria or taxa that are closer in the phylogenetic tree, they are believed to have a closer evolutionary relationship. Okay, then here I want to introduce some notations. So for the 3 matrices I have introduced here, we have the sample by taxon abundance matrix. Again, the taxon, you can think of it as just bacteria. So this is the microbiome sequencing data. And we denote it has n number of samples, m as number of taxa or bacteria, and the entries are log transformed read counts. So there are 2 ways to deal with this kind of data. Some people prefer to just use the row count data, and some people want to use the log transform data. So it's like basically the choice of the method developer. And here, depending on our model choice I will describe later, we prefer to use the log transformed data. And we also have the sample by covariates called sample metadata matrix. And we have a taxon by taxon pairwise distance matrix D that encodes the phylogeny information, basically the evolution information among different bacteria or taxa. So here is again a diagram that illustrates what we want to do. We will have the matrix to impute is basically the log transformed abundance matrices. And we will want to leverage the information from the sample covariates and the taxa phylogeny distance. We want to basically use these 2 information to impute and fill in the zeros that's in the taxa in the sequencing data. And if we look back at the sequencing data, we can see that already there are a lot of zeros here. And so the first step is to identify the values that need imputation, or we regard as non-biological zeros. So first, we can look at this is actually a mixture model. So you may think it's kind of intimidating, but I will explain step by step how we come up with this formula. And we also use the likelihood ratio test to check if the mixture model fits better than a single normal model to taxon J's abundances. And if the likelihood ratio test is significant, we will estimate the probability that Yij comes from a gamma component. So again, this seems kind of intimidating, and this is usually how we present our work to other people. But for the purpose of this seminar, I will explain each formula individually. So first, we construct a mixture model composed of the gamma distribution for processed false zero counts and a normal distribution for processed actual text abundance based on covariance. So for taxon J, we fit a gamma normal mixture model to its abundances across samples, and it looks like this. So this is the first part over here. And here, basically, you can, just from the formula, you can see that this is the PDF of the gamma distribution, and this is the probability density function of normal distribution. And we have some probability that the log transform reads will come from this distribution, and some probability that it will come from the other distribution. So first, what is the distribution? So the probabilistic distributions can be viewed as a mathematical function that with input as event and the output as the probability of that event. So the two distributions I have previously used are the gamma distribution and normal distribution. So the normal distribution with different parameters looks like this in the left-hand side, and the gamma distribution with those different parameters are on the right-hand side. So like I will explain later why we use these two distributions. But before that, I want to further introduce the probability density function. So a random variable x has a continuous distribution if there exists a non-negative function f, and the probability that x takes a value in the interval is as follows. So when x is greater than or equal to a, less than or equal to b, the probability of that is the integral of x from a to b. And the S described above here is called the PDF of X. So this PDF is defined only for the continuous variables. So if the X, or random variable, takes on the continuous values, for example, is defined on the real line, so it will have something like this. And if X is discrete, for example, if we flip a coin, it can only get a head and a tail, then that is called the probability mass function. So basically, we have a point mass at 0 when we got a tail, and the probability mass at 1 when we got a head, and each is just 0.5. So here is different. We have the X on real line, and we define a PDF in this way. And for example, the PDF for the normal distribution looks something like this. And we denote it as the PDF of the normal with parameters mu and sigma. Okay, so here is the example of the model fitting. So those are coming from the real data. And we can see that we have this bar corresponding to the probability of getting or the proportion of the values that are zeros in the real data for one taxon or for one bacteria. We can see in order to in our mind, in order to determine which part are the real, the biological zeros and which part are not biological zeros, we try to fit a gamma distribution and a normal distribution to separate them from each other. And so the choice of using a gamma normal mixture is based on previous literature. However, we can see that the distribution here really did fit well if we use the gamma and the normal mixture. And however, in some cases, we may fall into this scenario that if we just fit one normal distribution, it is good enough to capture the variance in the data. So if we fit a gamma normal mixture model, it may have a result like this. So then it's hard to decide which part of, although we can calculate the probability of the counts belonging to the gamma distribution, but it's hard to say that those counts should be not zero, because if we just use the normal distribution, it can well capture the variance in the data, basically. So in order to better identify the values that need imputation, we say that fitting the mixture model on a single mode counts. So this you can see there's one peak, so we call it one mode. So fitting the mixture model on single mode count data is not what we desired, and the processed counts belonging to a one mode distribution should not be regarded as false zero counts. So what we will do is to use the likelihood ratio test to check if the mixture model fits better than a single mode model to taxon J's abundances. Okay, so here is a point I forgot to mention, but I want to mention it again. So if we look at here, so this is the mixture model we want to fit for our data. So we see our data has two modes and we fit the mixture model. And here is the likelihood ratio test. You can see that the difference in the parameters is that there is actually an extra hat here. Oh, sorry, I made a typo here. This should be just gamma hat. So basically you can see every parameter has been added a hat over here. So this is the in the first equation, we are assuming the true model, or the model for the population. So the parameters are kind of we can think of as the true parameters we want to estimate. And after we plug in, or after we apply the model, try to feed the model on our observed data, then there's randomness, and we can think of that if we feed the model to different data, then we'll get different alpha hat and beta, we'll get different estimates of the parameters that best fit the data. So here we use we put a hat over that parameter to indicate this is estimated from our data. So you can see the difference. So here we have added the hat after we feed the data. Okay. So, okay, then what we want to do is to use a likelihood ratio test. So the second part to check if the mixture model fits better than a single mode normal model to taxon J's abundances. And so the likelihood is defined in this way. So L theta is our parameters. So basically everything before, you can see the alpha here, beta here, eta here. Those are all the parameters, and we denote it as theta. So you can think of it as a vector contains all the parameters we have, and x is what is our data. And so this is actually numerically equal to the probability of given those the parameters what we observed some values. For example, if we have a Bernoulli distribution, or if we roll a dice and we have the probability of get a hat to be 0.5, then we have theta is just a p, so denotes the probability of get a hat is equal to 0.5, and the px equal to 1, for example, to get a hat. So this is maybe just p. And so we write it in another way, is that we want to, what we really care about is the parameters. And we want to see, depending on the data we have, how we can write kind of the likelihood of the estimators. So yeah, so of the parameters, sorry. So that's why we write the likelihood. So you can imagine it's numerically equal to the probability depending on some parameters. What's the probability of getting a certain value of x? And in terms of the probability density function I have mentioned before, the L theta condition on x is just equivalent to the PDF numerically. And it's just kind of another interpretation. Here, we really care about is the estimation of the parameters. And the log likelihood is just the log of the likelihood, or the probability here. Okay, then the likelihood ratio test, so as I mentioned, is applied to select between two statistical models, the linear model we have and the mixture model. So the linear model is basically the normal distribution with the mean as some product of the parameters and the covariates. And the mixture model is the one we have with the combination of the gamma distribution and the normal distribution. So it's, yeah, so it's like this is the mixture model, and the blue line is just the normal distribution. Okay, so the likelihood ratio test is only valid for the model selection in hierarchical models. And so by hierarchical model is that for a more complex model, we can always set some of the parameters to 0 and to reduce to a simpler model. So if this is the case, those two models are called hierarchical models. And it's known that adding more parameters in hierarchical models will always increase the likelihood. And so here, when you add more parameters, you need to refit the model and guess the best estimators for your parameters, and then it will always increase the likelihood or the probability, which means the model can better fit the data, better. And the test statistic for the likelihood ratio test is like defined as what the name suggests. It's the likelihood ratio. So we have one likelihood and the other likelihood. The sup here means the maximum. So we want to find the theta that can maximize the likelihood. So basically, as I said, we need to re-estimate those parameters and get a best fitting from our model. And if that's the case, we have theta 1, which include more. So basically, then this statistic will follow a chi-square distribution. So if you recall, chi-square distribution is the third distribution I have mentioned so far. The previous two are the gamma distribution and normal distribution. So traditionally, when statisticians, they derive such distribution is like for the ease of hypothesis testing. I think Dongyuan have introduced you about what is the hypothesis testing, what's the p-value, and traditionally when statisticians do not have the computer, they want to derive some good distributions, so they have a table to refer to calculate the p-values or the probability of some value falls into that distribution. Okay, so this is the likelihood ratio test. So the key take-home message is that the likelihood ratio test can be applied to select between two statistical models in the hierarchical model setting. Okay, so then this is the plot I have put into my paper. It's about when we use the gamma normal mixture to feed the data. And you can see it has a quite good performance, I would say. And this is comparing with just feed data with one normal distribution. Okay, then in the last step of step 1, if we let the likelihood ratio test p-value equal less than 0.05, which means the more complex model actually is our model of choice. And if that's the case, we will calculate the probability of a count to be false zero by estimating this probability. You can see this is the ratio of the kind of the likelihood of the gamma distribution. The PDF of the gamma distribution divided by the sum of the estimated total. You can see this is the gamma distribution, this is normal distribution. So you can see it as a ratio of the probability densities. And so if it's greater than 0.5, we will say that Yij needs imputation. And if Dij is less than 0.5, we will say Yij does not need imputation. And based on the Dij's, we define a set omega of sample taxon pairs. So here the sample taxon pairs is, you can think of it as just the two indices in a matrix, so I and J. If you recall that in our matrix, the rows are samples and the columns are taxon. So one Ij is one entry in the matrix. And we want to see if that entry is actually needs imputation or not. So that's how we define the two sets omega and omega complement. And the benefit of doing this step is to convert the previous convoluted problem, which involves different kinds of zeros, and to a simpler problem. It's just a missing data imputation problem. And one famous example is
probably this is a good time to pause and ask if the students have any questions, because you have covered a lot of materials already. Probably
sorry about
before we talk about the Netflix.
Okay, so do you have any questions so far? Yeah, I just try to cover some of the materials like in probability and statistics that's related to the first part of my master. Yeah, in this talk. Okay, I see.
Vivian has a question in the chat box.
Yeah, I will read it out. So Vivian said, just want to comment that I recognized a lot of statistical methods you talked about from my AP stats class, like PDF and the chi-square distribution. Right, yeah, I think those are the fundamentals you need to learn during the undergraduate so that if you want to do future research, you will, okay. PDF is pretty much all I recognized from Charlie. Yeah, so basically, you can think of the PDF as to describe how the probability are distributed to different values on the real line. So it's kind of like that. Yeah. So are there any questions? So for Charlie, do you have any other questions regarding, so which part do you think you may not understand very well, or do you want to say that?
If I'm being completely honest, PDF is truly the only thing that I recognize here.
Okay.
And I think the graphical methods are like those help me understand a little more than like the equations, because the equations are just a bunch of like parameters that I'm not.
Yeah.
But
then are you familiar with like the normal distribution? So it's the bell-shaped distribution that's how usually used. And I think maybe the gamma distribution is a little bit like maybe you did not learn it yet. You haven't learned it yet, maybe. So it's not just, yeah, so you can think of it just how the normal distribution works, but it has a different shape and a different kinds of parametrization. Yeah.
So like the reason that it's different is because of different parameters?
It's like they are derived in different ways and they have different like meanings. For example, the normal distribution is like, so are you familiar with the binomial distribution? Yeah. Like the sum of independent coin throws. Yeah. So like I think in some cases, if you like throw a coin very many times, and if you count the sum of heads, and that will be the binomial distribution. And if you do it kind of infinitely, it will converge to the normal distribution. And for the gamma distribution, they have some like other kinds of meanings behind that. So like statisticians kind of derive it from other perspectives.
And so every common distribution has a reason to be developed or proposed. So if you're really interested in those, I would suggest that the Stats 100B is a very good class that covers all those fundamentals Ruochen used.
Yes.
Mm-hmm. Yeah. But I know many students would think the 100B is a pretty tough class. Yeah, I heard people say that. And I taught it for several times myself, but I really want to emphasize that it's very important if you want to be able to understand those statistical methods. So that's just required. Otherwise, you would just be able to use the R packages using command lines, but not really understanding what's inside.
Yeah, I think, and also those distribution and statistical concepts, a lot of them are related to gambling or such things. So it's kind of interesting when you look into it.
Yeah, gambling is about discrete distributions like binomial, negative binomial, those kind of things. And these are continuous distributions. And most of the time, I think continuous distributions are much more difficult to understand because you need to have some concepts about how real numbers are like before you can really understand the continuous distributions. So to put it in a simple way, gamma distribution has its domain, which means the possible values you can draw from a gamma distribution on the positive real line. So you can never draw a negative real number from gamma distribution, in other words. While normal distribution, in theory, covers the whole real line. So you can have negative value, positive values, but it's very concentrated around its mean. So we know that if you take, say, 3 sigma, 3 times the standard deviation from the mean on each side, then in that interval, you can capture 99% of the chance you will draw values from that interval. So even though theoretically you can get positive infinity or negative infinity, but in practice, you will never get it. So that's just too small probability. Yeah. So here, in Ruochen's model, he chose the normal distribution as the second component on the right, just because of the shape of the data. So it looks like a bell shape. And also another reason is normal distribution is commonly assumed for linear model for convenience. And also another reason is a lot of the noise we observe in daily life follows normal distribution. So that's why in linear model, the random noise term is assumed to follow a normal distribution, and Ruochen borrowed from that. But he added a gamma component on the left to explain those small values that couldn't have come from a normal distribution. So putting them together, he can explain the shape of the data he observed.
Yes.
That makes sense. Okay, thank you.
So that's basically what I have done in the first step. And so after doing that, we like convert our problem into a missing data imputation problem. And one famous example is the Netflix data challenge. I think maybe Kexin has talked about it. So like for some people, they have ratings for movies. However, for some people, they do not have ratings. So how can we infer what the ratings he will have for that movie, depending on both his previous ratings and other people's ratings? So that's the kind of summary for the Netflix data challenge. So then in our problem, we try to impute on the entries. We think they are probably not due to biological reasons, but due to technical reasons. And in order to do that, I want to borrow the information from both across samples and across taxa. And so you can see here in this cartoon diagram, I picked out this column and also this row, and I want to borrow the information from the other cells. You can see from those entries and to impute on that value. So this will be raised to a higher value after imputation. Okay, then in more detail, so if you recall that omega sample taxon pairs whose abundances are unlikely missing and thus do not need imputation, and the omega complement are sample taxon pairs whose abundance need imputation. And our imputation assumes the linear model. So it's the similar taxa across different entries and the similar samples across different taxa times some parameters. And also we want to borrow the covariance information. So if you recall the second matrix that contains the BMI, the gender of the samples, we want to also leverage that information. And we add a epsilon is a Gaussian or normal random noise. So basically this term will follow the normal distribution. Okay, and so okay. Then this is a diagram for the imputation. So it's I'll try to make it easier for you to understand. So basically it's still the same thing that is for each entry. We will borrow the information across different taxa and across different samples. And we also want to leverage the covariance information to predict what the value should be that's missing. Okay, and right now the number of parameters we have, if we calculate it, it's m is m squared plus n squared then, however, training sample size is just the entries that are not that do not need imputation. So this is of course less than or equal to the dimension of the original matrix, which is m times n, because you can see it's a subset of the m times n entries. It's just a subset that do not need imputation. And this is called a high dimensional parameter estimation. And I have constructed this penalized loss function. So here the DJJ prime is just try to utilize the previous mentioned third matrix, the taxon evolutionary relationship matrix or phylogenetic tree that I want to leverage it in the penalization. So so for now I will leave this formula like this, and you can just see through it. So basically it's like a squared norm between the Yij, the entries, and also the things we want to borrow the information on, and we want to penalize it on those parameters. And so here, what I mentioned, the concept is called a high dimensional problem in linear models. So if there are more features than observations, we will run into the problem that the model will be able to perfectly fit the data. This problem is known as the model overfitting, and one consequence is that the model is not generalizable to new data. Another problem with a lot of features is that the final model will be hard to interpret. And there's one way to deal with this problem is to penalize on the value of the parameters and aim to shrink the close to zero parameter estimates to zero. And so here is a very popular approach called least absolute shrinkage and selection operator called lasso or lasso. So some people read it as lasso and some people read it as a lasso. So basically it's LASSO, you can just remember it. And so based on the linear regression, the penalize L1 norm of parameter, they penalize on the L1 norm of the parameter estimation. So if we let beta be a vector basically of d dimensions, then L1 norm is defined in this way. So basically you can see that beta is a vector. So it's the L1 norm is just the sum of its absolute values in each dimension. And the L2 norm is defined in this way, that the L2 norm of that vector is the square root of the sum of the squared of those of the entries in each dimension. And the lasso puts a constraint on the L1 norm of the parameter estimates and gets the good property of getting a lot of parameters shrinking to zero. So as I mentioned, if we have a lot of parameters shrinking to zero, which means we only left with a very few parameters that have non-zero estimates, that will avoid the problem of overfitting and also has the benefit, and we can select which features are more important in our model. And do anyone, I think this is kind of straightforward. So basically the L1 norm is just the sum of the absolute value in each dimension. So I think, yeah. And so here are two plots that shows why that is the case, that the L1 norm will induce a lot of zero values. So here we can think that if we try to use L1 norm, so first, if we look at the beta with only 2 dimensions, which can have beta 1 in the first dimension and beta 2 in the second dimension, and versus, so if we had put a constraint on that, so if we let the L1 norm equal to some constant, then it will form a line in the 2D plot. Basically, we can have, for example, A times beta 1 plus A minus 1 times beta 2 equal to C, and that will form a line on this plot. However, if we look at L2 norm, then we have the beta 1 squared plus beta 2 squared is equal to some constant, and that is a circle. And if we have some objective function that intersects with those two constraints, we can see that for the L1 norm, it's more likely to have a zero in that dimension. And so if that's the case, then it will basically shrink the parameter to 0. So those two graphs that show the similar idea. Is just that on the right-hand side, the objective function is kind of a circle, and on the left-hand side, it's just a line. So I think if you like later, maybe if you want to do research, you may encounter this kind of plots when you deal with the penalized regressions. Okay, so then I will talk about the results. So first, I want to evaluate is how accurate is our imputation algorithm. So here I used two metrics. One is the mean squared error, and the other is the correlation. This one is actually the average correlation among all the taxa. So I will explain it in a second. But basically, the lower the MSE, the better, and the higher correlation, the better. So you can see in both cases, our methods have a better performance comparing to some kind of imputation methods in single-cell RNA sequencing and the soft impute, which is a very widely used method, I would say, in machine learning field. Okay, so in order to evaluate the imputation accuracy, so in our simulation, we first simulate a matrix Y, which we call the complete data, and then we introduce zeros to Y and simulate Y prime. Finally, we perform imputation methods on Y prime and get Y hat. So, and after that, we compare between Y and Y hat to evaluate the simulation results. And the mean square error is defined as follows. So basically, you can see that you can think that the Y is a matrix, and the Y hat is a matrix of the same dimension as Y. And so for each entry, we measured its the squared difference of those two matrices, basically, and we average it by the total number of. Actually, I shouldn't write N here, but that's the definition just for MSE. So in our case, it's actually M times N. So it's the average of the square distance between every entry in the complete matrix and our imputed matrix. And the Pearson correlation between two taxa is, so here is the sample Pearson correlation is defined in this way. If you are kind of familiar with the statistics, it's just the covariance divided by the standard deviation of Y and the standard deviation of YJ hat. Yeah. And so this is the Pearson correlation between two taxa, and we then calculate the average of those correlations among all the taxa. So these two measures result in the plot I have been plotting over here. And then to further evaluate the imputation accuracy, I look at the so this is the taxon mean divided by taxon standard deviation. And so this is the complete data simulated. This is the, after I introduce zeros, the taxon mean divided by standard deviation looks something like this. And after our imputation methods, the shape of the taxon mean divided by standard deviation looks like this. And those are for the other method. And we can see that our method is most similar to the complete data. And so here I use a D, is called the Weissenstein distance. And so the smaller the distance, the better. So our method has the smallest distance with the complete data. And also I plot the taxon mean versus taxon SD. And we can see that this is the shape in the complete data. This is when we introduce more zeros into the original data. And this is what has been recovered by our method, and those are for the other method, and our method is still better. So in order to evaluate those accuracies, so so the goal here is to argue that the imputed data by mbImpute better resembles the complete data comparing to other missing data imputation methods. The Weissenstein distance between the distribution of taxon mean divided by taxon SD of complete data and the imputed data. And we use the Euclidean distance between the mean and standard deviation for taxa in the complete data and the imputed data. Okay, so yeah, so basically the Wasserstein distance can measure the distance between different distributions. So we can kind of see here, we can draw the distribution of taxon mean divided by standard deviation in each scenario. And we want to measure how those, you can think of as measure how those shapes are different from each other. And that's is, we can use the Wasserstein distance. Okay, then I performed other analysis. For example, the identification of differentially abundant taxa. Basically, I perform a statistical testing and see that what taxa have different abundances in control samples and in disease samples. And like after, so those Wilcoxon t-test and comes in D6-2 metagenome-seq, those are like some commonly used DA methods in the field. And after imputation, we are able to make them again in the precision, recall, and F1 score. So basically, the higher the bar here, the better the result is. And we compare it before imputation and after imputation. And we think that our methods can facilitate those DA analysis. And here is a plot that shows after imputation, our method better resembles the shape of the original non-zero distribution comparing to the soft impute. You can see that the soft impute has some unusual high peaks. However, our method will have like similar shape comparing to the original non-zero values. And then for the real data studies, I evaluated how so like before, like between different data sets generated for the same purpose. For example, they all are interested in the type 2 diabetes, and they generated 4 different sets of data sets. And I want to see if there's overlap in the findings. For example, in the differentially abundant taxa that can be detected in all of the 4 data sets. And so here's the result. It shows that after imputation, we have more common findings comparing to without imputation. And I also evaluate the cross-study sample prediction results based on the identified DA taxa. So this is like when I use the one method to find the DA taxa, and I perform prediction on the other data, just use the DA taxa I found in the previous data. So to evaluate what's the classification accuracy. And we can see that mbImpute can even let the can make the subset of the matrix gain a better prediction result. And so here is the plot I shown that if we only look at the correlation between the non-zero entries. So this is the correlation between non-zero entries, and the black line is the correlation between if we don't omit the zeros. So we can see if we have a lot of zeros on at zero values for this taxa, then the correlation may be dragged by those zeros. And so you can see there's a large difference between the non-zero correlation, the correlation of the non-zero data, and the data with a lot and the whole data. And after we perform imputation, so we can see.
Sorry, yeah. I think you didn't say it correctly. You said correlation between non-zero data. That's not right. This is the correlation between 2 taxa, between 2 bacteria species. Just how you calculate the correlation. Did you only use the non-zero counts, or you use all the counts? If you only use the non-zero counts, the line between the linear regression line between the 2 taxa will look like will be the blue one. If you use every data point, it will be the black one.
Yes, right. So basically here the results is to show that after imputation, the correlation the correlation between different taxa we can get more resembles the non-zero the correlation between taxa, just use the non-zero entries. Okay, and also, yeah, so maybe I can skip this one and to the summary. So in summary, mbImpute is an accurate and robust imputation method designed for microbiome data. And it corrects the non-biological zeros and retains taxa's non-zero abundance distributions. And we did utilize the sample metadata and the taxa phylogenetic information for the imputation. And it outperforms the imputation method developed for other data types. And it can empower the DNA analysis and facilitates the network construction of microbiome data. So I have written an R package for this, and the paper is currently on bioRxiv. And so I want to acknowledge my advisor, Jessica, and the collaborator, our former PhD student, Vivian Lee, who is right now an assistant professor at Rutgers. And so those are the computation methods I have been comparing with. So that's all for my presentation, and thank you. And feel free to ask any questions if you have.
Thank you. So I think before or after we paused and asked the students to ask questions about the densities and the likelihood ratio test, those backgrounds, the second part was about Ruochen's results, the comparison results and the evaluation results. So I think probably this is something easier for our students because they do not require so many technical backgrounds. Instead, you can just think from a high level, how did you evaluate an imputation method works well?
Yes.
So probably Ruochen can explain what each figure is like, or what's the name of the figure, why do you use it for illustrating the results?
Yeah, so the first type of figure is called a bar plot. So each column here is just corresponding, so you can read out the value of that from the y-axis. So basically here the MSE for the first method without imputation is around 4, and after we perform mbImpute is around 0.8, and for soft impute is around 1.5. So you can read from the bar plot about the values here. And then here is the histogram. So the histogram are a good way to visualize the distributions because the probability of a value between those two, the left endpoint and right endpoint has been shown here. Although here I didn't show the proportion, I just show like how many values are between those two, but you can normalize it by the total number of points here, and you can get a probability. And so that's what histogram is for. And it's a good way to visualize what the distribution look like. And this plot is called the scatter plot. We have the x-axis and the y-axis. So here the x-axis is the taxon's standard deviation, and the y-axis is the average value or the mean value of each taxon. And we can see for each point here, it corresponds to one taxon. And so this is basically see what's the shape, what's the relationship between the taxon's mean and taxon's standard deviation. And we found that our method resembles best the complete data comparing to other methods. And so here, this is again a bar plot, and we can read the values directly from the y-axis from the bar. And this is again a histogram, but yeah, it shows just the shape of the like the raw data, and after our imputation, what the shape of data looks like. This is again a bar plot, shows the like the prediction kind of sorry, this is a bar plot, and you can read out the overlap in findings between different data sets. Yeah, this is again a bar plot, and this is again a scatter plot that shows what's the relationship between two taxa, and we evaluate the correlation based on only on non-zero values and on all the data. And if that's the case, we also, so here actually this line is a regression line, so we perform a major access regression, and so it's a technique, and basically it's a linear it's a line that shows what's the relationship between two between two taxa. And this one, okay, so I think this one I will omit this for now. Yeah.
Okay, thank you, Ruochen. So do you have questions from our students? Maybe you can ask Ruochen if you have any questions regarding the microbiome data. So why do people generate them, and how can those data be useful? Because Ruochen's talk is the first one so far that is not about single cell data. So this is a whole new application domain.
Yeah, I think you may heard about some like kind of yogurts that talk about how the microbiome, like the bacteria, can affect your gut and make you more healthy. Although that may not be correct, but there are scientific research shows that your health is closely related to microbiome. So related to the bacteria that lives on human body or human gut or in the environment. So basically, you can think of the interaction, the interplay with the microbiome and the environment, and so they are interacting with each other. So it's like very interesting to study how they really affect people's health, especially in some certain diseases. If they can find a kind of a relationship between the bacteria and the disease, they can introduce, for example, a new kind of bacteria, or try to use some medicine to kill some of the bacteria to adjust your health, basically. And the sequencing is just one approach to measure the abundance of the bacteria in a certain environment.
Roger, maybe you can say a little bit about how we were motivated to do this based on our previous work that imputes the missing or the abundant zeros in single-cell RNA-seq data.
Yeah, sure. So previously, our group has published a paper called mbImpute. You can see from its name, it's quite similar to this method, but it's like based on the single-cell RNA sequencing data. I think you may have already learned a lot in the previous talks. And so in the single-cell RNA-seq data, because the RNA content in each cell is very low, so it may lead to a lot of zero values in the RNA-seq data. So at that time, our group was among the first who tried to use imputation to address this problem. And you know, in the common statistical settings, usually the data statisticians or practitioners collected are not so sparse. It will not contain so many zeros. And so the traditional statistical testing or modeling are all based on those data that may not contain so many zeros. Right now, due to the sequencing kind of artifacts or due to this phenomenon, we can see there are a lot of zeros in the data, and that may affect our use of different statistical methods. And by using imputation, we borrow the information and we make the data less, make the traditional methods more applicable. And some people have shown that the biologists will see that those data are like resemble what they've seen before. Like before the single cell sequencing data was introduced, there was bulk sequencing data, and the bulk sequencing data also contains less zeros. And so the the biologists has already made many discoveries based on the bulk sequencing data. And when they see that the single cell RNA-seq data after imputation, they resemble the bulk RNA-seq, or the single cell RNA sequencing data, they resembles the data from other kind of single cell analysis technologies, such as some method called FISH, or they may think that after imputation, it carries more biological meaningful information. So that's kind of the background for our last paper that performs the imputation on the single cell data. And then I think at some time point, a very famous statistician in the microbiome field has visited UCLA. His name is Hongzhe Li, and he talked with Jessica about the idea of try to use imputation in the microbiome sequencing data, because he thinks in the microbiome sequencing data, we are facing similar problems of having a lot of zeros. And we want to try if the imputation works here or not. And when I approached this problem, I found that different from single cell RNA-seq data, the microbiome data also have the covariance information for the patients, and also the phylogenetic information that encodes the evolutionary relationship among different taxa. So I developed this method to try to incorporate as much information as possible in order to provide a reasonable imputation method for microbiome data that can facilitate the biologists to make new discoveries, basically.
Thank you, Ruochen. Okay, so Ruochen has said a lot. So yeah, let's wait for questions from our students. As always, any questions, no matter whether it's about the background or specific to a plot, all of them are welcome.
So now that you've kind of developed working with these imputation methods for these types of studies, is now the focus on like improving those methods, or is it finding newer methods?
That's a good question. And I think actually our method was the first method that tried to devise the imputation for microbiome data. So maybe if you like, you could try to develop more imputation methods. And I would say, like, if some people find it useful, they will apply my method and see if they can find more biological meaningful findings. And I think the ultimate goal is still about finding more like interesting findings that can help, for example, the doctors to kind of diagnose or to treat a disease. So that's still the final goal. The statistical method is kind of a step that can help them to do that. Yeah. And so basically, what kind of research you want to do is basically on your choice, and you can see whether you more like to develop new methods or you are more interested in biological discoveries. Did that answer your question?
Yes, thank you.
Oh, yeah.
Ruochen, maybe you also want to say that you want to investigate some properties of different imputation methods, or in general, the general imputation method that can have broad applications, but fundamentally they use different mathematical techniques. And that's your next project. You want to compare the methods.
Yeah, okay. So here you can see in my formulation, I formulated it as for the, basically it's a linear model. So for the value for each entry, I assume it's a linear model, so it learns from different taxa and from different samples. And however, there's, like, as I mentioned in the Netflix data challenge, the most successful method is about the matrix factorization. And the so-called the method I compare with, the soft impute here, that one is a very famous method, and this this one is also based on matrix factorization. So I'm interested in learning what's the difference of the kind of regression-based imputation methods and the matrix factorization-based imputation methods. And I want to see which methods is more, for example, more successful in what way, and about, for example, the computational efficiency, and also about some kind of statistical properties to compare between those two types of methods.
Okay, yeah, so I think Ruochen has really explained his work in very great detail, and a lot of you have, I think you might have a lot of questions, but that may not be easy to formulate at this time. And so maybe Ruochen, can you help me by giving a brief preview of what we will have in next quarter, just as a preview announcement? I think you were helping me try to find the speakers, right?
Yeah, I think Yiling and Xinzhou will talk in next quarter, but I don't know what they plan to talk about. Probably about, yeah, I'm actually not sure about what their topics are, but you would imagine that will contain single cell, about single cell, about the traditional statistical methods. I would say just a lot of fun in the next quarter. So I would highly recommend that if you could, you can take it again and see what other students are doing in our group. Yeah.
Mm-hmm. Yeah, and also I can welcome some outside speakers as well to give you another aspect or perspective about what's out there in this computational genomics field. Like my group focuses a lot on the statistical part, but other groups at UCLA, they focus on more computer science, which means that they focus more on algorithm design and computational efficiency. And there are also biology labs that use computational methods, but they do not develop methods. So their focus is more on the biological question to be answered. So although we all sit in this intersection, but we have different focuses. I think that's also something good to know for our freshman students. Okay, so if there are no more questions, I think that's all for today. And so thank you all for being here this quarter. And I know that this should be a busy week, right? The end of week 10, you're going to have a final week. And this is tough because we're doing the remote education, right? This is your first year at UCLA, but you just ran into this remote teaching. So hopefully we will welcome you back to the campus next year. But for next quarter, so this course will still be remote and recorded. So if you have any questions, please feel free to let us know. And also, I will greatly appreciate your evaluation for this class. I think we still have evaluation likely. So I think the UCLA has announced that they have some technical issues with the system. Like they fixed it, but they extended it, and then they extended the deadline. So you have a few more days. Previously, I think the deadline should be tomorrow night. So that's very soon, but you will have more days to do it. So thank you all for your attendance, and also thank Ruochen for his talk.
You're welcome.
Okay, goodbye, everyone.
Thank you very much.
Thank you. Thank you.
Thank you.
Thank you.
Thank you.
Thank you.
Thank you.
Thank you. Okay, so I think that's all, Ruochen. Very good job.
Thank you.
Okay, bye.
Bye.
Bye.